Question 2. Figure 1 shows some of the stages in the production of human insulin by bacteria. The stages are not in the correct order. And we can see there figure 1 on the right with A, B, C and D. I always think it's really useful to look at the diagram before you even look at the rest of the question. Just make sure you're clear what all the keywords mean, what it's telling us. Okay, after we've done that, we then look at the question. So write down the order in which the steps should take place. The first one has been done for you. We can see D has been done for us. If we refer over to figure 1, we can see that D is showing us the insulin gene being removed, being cut out of the human DNA. So therefore, we're thinking to ourselves, okay, what is going to be the next stage in this process? Well, we've got the insulin gene. We need to put it into a bacterial plasmid. We can see that happening in A. So A is the next answer. We're then thinking, okay, what do we now need to do with this bacterial plasmid? Well, it needs to go inside a bacteria. So we can see that happening, going into a single bacteria in B. Then finally, we clone that bacteria, we cause it to divide. We can see therefore that each of the clones of the bacteria then has the plasmid with the gene in, and we can see that happening at C. Those 3 correct letters score 2 marks available for this question. For part B, we're told that insulin is used to treat people with diabetes. This insulin used to be obtained from animals. An advantage of using insulin produced by bacteria is that it does not involve killing animals. Explain one other advantage of using insulin produced by bacteria. Now, it's very clear here that it asks us for one other advantage. So you can't put anything about animal cruelty or killing the animals. So it's got to be something else about the insulin being produced by bacteria. We can also see that it's a 2-mark question. So any answer we give must be developed, sort of explaining a reason why there's an advantage there. It's not just state the advantage, it is explain the one other advantage. So the correct answer here is it is identical to human insulin, as in the insulin produced by bacteria is identical to human insulin because it's using the human insulin gene. Now that then gets us 1 mark in this question, the fact that we say it's identical to human insulin. The second mark comes from explaining why that's an advantage, and it's that it is less likely to be rejected when it's actually injected into humans. So when it's used in humans, human insulin produced by the bacteria is much less likely to be rejected than one from another animal. So that second explanation there gives us our 2nd mark.